Right. Welcome to the last mathematical biology seminar of this semester. Today we have Sophie, and she will share her study about the nanoscale caterpillar, or how to move precisely with random sticky feet. Thank you, Sophie.
Thank you, Kana. I guess since it's not a lot of us, don't hesitate to bump in and ask more questions. This is work I've I've started during my postdoc and that I'm continuing a little bit, and that I thought would be most relevant to this crowd. So to give you a coarse-grained picture first of my work more generally, let me start by something which is very common to each and every one of you, which is coffee. And you know, every once in a while, it happens that we take a spoonful of sugar, dip it in the coffee, and then use the spoon to stir the sugar inside the coffee. This experience of mixing at the macroscopic scales, it's very common, you know, at our scales, but at the very small scales, which are interesting for me, there's no such thing as a nanoscale spoon to achieve a complex task such as mixing, but not limited to mixing. You might also think about sieving, selecting, and displacing specific particles in some place. Now, there's something that's fascinating to me at these very small scales, and that's the presence of mechanical fluctuations. Oops, let me jump straight there. What I mean by mechanical fluctuations is that these small scales, everything is jiggling around. And what I'm hoping to find is ways to harvest these mechanical fluctuations to achieve these specific tasks of mixing, sieving, selecting, and so on, that I call tasks of macroscopic transport. Now, throughout my research, I've been looking at this question in different ways and on different systems. One of the first places where I started thinking about these things was in very small pores, for example, in nanoscale pores that are like ion channels, for example, in biology, where the impact of surface fluctuation dramatically affects the transport within the pore. And so this is something I started studying during my PhD, but that we're still having questions on. And then when you think about little pores like this, ion pores, at some point, you only have a few molecules or a few ions at the same time inside the pore. So there are important fluctuations of how many particles you have in there. And this has consequences in terms of how specific you can be in terms of detection, noise signals, noise signatures, and so on and so forth, which are a topic of activity for me right now. And then today, I'm a little bit going to reverse the problem and talk about, the source of these fluctuations all came because I was looking at confined things. And so the fluctuations came from the confinement or the place where you were in. But in fact, you could think about the object under study, so here a particle, to have lots of fluctuating degrees of freedom. And that's what I'm going to talk about today. So I will tell you more in a second about that. And then right now I am orienting a little bit my research line into understanding how fluctuations in living soils affect dynamics in soil, reaction kinetics in soils, and so on and so forth. So if you're into degradation, metabolism, transport inside soils, please let me know. I'd be more than happy to chat about this. Okay, so today I'm going to talk about fluctuating sticky feet particle. And before I go deeper into the subject, I'd like to thank the people with whom I did this work. So Gina Zhang, Fan Kui, Dave Pine, and Miranda Holmes-Serfon. And this all started when we were all back at NYU. Okay, so particles with sticky feet. What are these things? So I like to call them nanoscale caterpillars, and I think you'll get the image in a few seconds. So what are they? They are particles, so I draw them with this yellow sphere. They can be cells or colloids, and I'll go back to examples later. And these are particles at small scale, so a few nanometers to a few microns, so they diffuse. And then on those particles are coated a bunch of legs that are made of polymers of proteins. And because they're made of these jiggly materials, of course, there's thermal motion and those legs diffuse. And at the tip of those legs are specific molecular stickers that can bind and unbind to other specific receptors, for example, on a nearby surface. Now, the whole stochasticity of binding, unbinding, leg diffusion, the overall particle diffusion creates an overall motion of the particle that is non-trivial and influenced by all these little degrees of freedom. Okay, so I like to put this big mathematical framework on a category of particles that have many applications, in fact, and both in biology and in the artificial realm. And I'll bring you through specific examples of these particles and the kind of questions, physical questions that I think are that are interesting to me there. Okay, so in biology, there's one example I'm sure you're all familiar with, which is that of viruses. So viruses are definitely sticky feed particles. They have spike proteins that allow them to bind and unbind to specific mucus proteins on surfaces. This representation, for example, is a representation of the influenza A virus and of the experimental understanding that we have, but though not the detailed proof, visual proof of how it moves. So performing some form of crawling and gliding, and specifically in the study that I reference here, they find that when they tune the strength of the protein to spike protein bonds, the motion is clearly altered. So the detailed, dynamics of binding and binding definitely affect the motion of the virus, and then ultimately how the virus invades its host. So the biological process underway. So that's one question that is, for example, interesting to me. So viruses are kind of the bad guys, but there are also the good guys in biology. Those are, for example, white blood cells. So here I'm going to show you a movie of white blood cells. So these are these pink balls rolling inside blood vessels, and they roll at the surface of these blood vessels by selective binding and unbinding of specific proteins to the blood vessel wall. And ultimately, at some point, though it's not shown in this movie here, they will stop, and then they will permeate through the vessel wall and then, treat their host. So again, here, the specific dynamics of adhesion of these little proteins determines how the white blood cell rolls, and then how ultimately it stops, and then decide that it needs to go there and heal, that it has reached its healing target. And again, here we have many questions on how we relate those specific parameters to the biological process. So those are two examples in biology. And there are many, many more that I'm sure you can think of. And if you have more ideas, I'm happy to run through them. But there's also one I'd like to mention in the artificial realm. And it's one example of artificial materials called DNA coated-colloids. So those are colloids. They can be, for example, plastic or other kinds of material, little spheres from a few nanometers to a few microns in size. And on those colloids, there has been significant progress in sticking DNA. So what we stick is single-stranded DNA sequences that have their complementary DNA sequence that allows them to bind and unbind to specific other receptors, either on another particle or on a surface. And the kind of things that we've been interested in doing with these particles are typically artificial materials, because you can bind lots and lots and lots of these colloids together. And those materials have interesting light absorption properties, that being one of the examples, but there are many, many more. But to design these materials on a large scale, critical parameters in the experiments are required to form such assembly. So here I'm showing a movie of micron-sized DNA-coated colloids slowly aggregating into a crystal. And so you can start seeing crystal blobs forming, and then ultimately the crystal blobs try and merge into each other. But that's a tricky thing, especially you can imagine that if the bonds are too sticky, then you won't be able to heal this discrepancy between the two lattices. So critical parameters are required to form long-range alignment, and in fact, we don't really know how to be quantitative in which parameters we choose and why. So again, the underlying binding dynamics control here the self-assembly process. So hopefully I've convinced you and motivated the question that I'm interested in about these nanocaterpillars is, can we understand and then ultimately can we control their motion from the microscopic understanding? And to answer that question, today I'm going to try and go through two items. The first one is, well, if the nanocaterpillar moves, can I first of all quantify how fast it moves? And then can I understand, well, how it's actually moving? Okay. Any questions so far? Yeah, good. So today, to make progress, I'll be working when I show experimental data on a model system made of polystyrene DNA coated colloids that are polystyrene particles about 1 micron in size, coated with lots and lots and lots of single-stranded DNA strands. And those particles are coated with one sequence, and the complementary one is on the surface, such that we can observe the particles moving on top of the surface. Now, this is a schematic, and it's really not to scale. What you should have in mind on these kinds of systems, including some of the systems we saw in biology, is more something like this, which I showed at the beginning, where the first thing you might notice, where here the representation is this colloid as this blue sphere, and then the yellow hairs represent the DNA strands. Now, this image is to scale for a slightly smaller bead because it was impossible to generate something much bigger, but it gives you a better idea, I think. And I hope the first thing that's striking here is, wow, that this little guy is pretty hairy. And what this means is there are lots and lots of degrees of freedom, so basically of legs, that my particle can move with. So that's a technical challenge to solve, especially in modeling. And then another thing that's striking is, of course, there are very different length scales. So there's these small legs, the colloid is much bigger, and then at the end of the day, the route that the colloid will trace is even bigger. So there's numerous length scales at play. And then these little, shiny balls that I put in kind of represent the bonds. And the bonds here are chemical bonds, which means they're highly sensitive on temperature. And that's, the fact that they're sensitive on temperature is a challenge, but it's also a cool thing because it means experimentally tuning temperature is quite easy. So we can probe the system in different conditions. So how does the system react to temperature? Well, if I start my system at observing it at a rather low temperature, but it can just be, room temperature, all my particles are stuck on the surface, are bound on the surface. And then as I increase the temperature around anywhere between 25, 60, or 75 degrees, we can arrive at a point where we can detach all the particles from the surface. And so there's a sharp specific temperature that is called the melting temperature in the community that can be tuned anywhere between 20 or 70 degrees, but in fact, it can be tuned even wider, but we were interested in temperatures that were relevant to us. So anywhere between 20 and 70, where this transition from strongly bound to unbound happens. So I'll be looking at my systems all the time with respect to this melting temperature, and because it allows me to constrain a little bit the problem. Okay, so then what do? We start measuring, how fast it moves. So we do standard microscopy experiments where we look at our particle from on top. This is an image of a particle of 1 micron in size. We can track it with time. It performs a random walk. And we can track it at a given temperature, but we can track it also at lower and lower temperatures. And here I show, for example, a movie 6 degrees below the melting temperature. and one at the melting temperature. And you can see, hopefully, the movie right on the left, the particle is moving, is barely moving to the eye. It's moving a teeny bit, but it's barely moving compared to the one at just 6 degrees at a temperature that is just 6 degrees above. So of course we can quantify that. For example, we can quantify that in terms of the diffusion coefficient of our particle. And we find the following thing, that the diffusion coefficient of a particle relative to this relative temperature, relative to the melting temperature, spans 2 orders of magnitude in just about a few degrees Celsius. So those are all the experimental data points, and the slightly different colors are just for repeated experiments from the start. Okay, so we have a 2 order of magnitude change in the diffusion coefficient. Now that's a very big change, and we really wanted to understand why and how big. So to make progress, I started building a model to understand how fast it was moving. And then it struck us that to build that model, we really needed to go one step further than what was available in the literature for the following reasons. One of them is some models that were out there, for example, had no stochasticity in them, but we were measuring diffusion, which is inherently a stochastic process. So we needed something where noise was in there. Then some advancements were done for only just 1 or 2 legs, but we had, thousands of legs, and that's also the case for other systems too. And then one tricky, say, technical thing is that most techniques available were used for a small perturbation, but here we had a 2 order of magnitude decrease, which is not a small effect; it's a big effect. So we needed tools to get this big effect. So we built a microscopic model and trying to solve these issues. And I'm going to walk you through this model and basically give you the minimal ingredients that we put in. Okay, so this is the starting point of the model. We are looking at a particle that I represent by this rectangle, and the particle's position is written x, and it moves along a surface. And I'm interested only in lateral motion across the surface. So for now, I drew my particle as a square and not as a circle. So my particle, when it moves, it basically is diffusing with a bulk diffusion coefficient d naught, which is given by an Einstein law, kT over gamma, where gamma is the friction, the hydrodynamic friction that is due to the surrounding fluid. So I can write, for example, an equation describing the overdamped motion of my particle here to the bottom left. Now, of course, my particle is not just a particle, it has a leg attached to it. And I model that leg by a spring that represents well polymer dynamics, for example. And that spring has a given length, little l, that has a spring recoil force given by a spring constant, little k. And again, that leg is also jiggling and diffusing around with a friction coefficient that I write little gamma. So that's my small friction. Okay, now my leg can bind and unbind to the opposing surface to specific receptors. And to make the story a little bit simpler, I'm just going to consider a smooth, sticky surface where my leg can basically bind anywhere. It binds with binding rates Q on and an unbinding rates Q off. And the ratio of the 2 rates gives me the energy of the bond. So the specific chemical energy of making the bond. Okay, and now once my bond is made, when I try to move the particle, I also have to move the leg. They basically move. There's a kinetic constraint in moving one and then moving the other. And that means that when I try to write the equations in the bound state that rule my motion in the bound state, I have to be really careful, and I cannot just write anything. I have to project my equations, which is sometimes a step that is overlooked. And once I take into account this constraint that says that my leg needs to move like the particle, which is this condition at the very bottom, dl over dt equal minus dx over dt, I get in my equation of motion now 2 noise terms, one that comes from the leg and one that comes from the particle. So basically, both individual noises feed into the overall motion of the ensemble. So that makes something, a little bit tricky already. And now I have, basic ingredients. For now, I know that I have just one leg. I'll go to multiple legs right after. But just to show you the key things that go into the model. Now, what can I do with that? I can simulate that thing. It's not too hard, but it becomes pretty involved because I already have 5 parameters. So my parameter space starts to be really big. And then, of course, there are dynamics that are really fast, like binding and unbinding and the recoil force. But the overall diffusion of my particle is pretty slow. So I have to simulate for, lots and lots of time scales to obtain interesting behavior. So to go one step beyond, we used mathematical tool to get the macroscopic behavior of these equations. And the tool we use is called homogenization, where we basically where we basically properly averaged over all these very fast binding, unbinding, and recoil effects of the spring to obtain the overall motion of our particle. So here, I wasn't uncertain what you would be interested into, but if you want, we can come back to what goes into the detail of that. But for now, I thought I would rather, continue on with the understanding that we got, but I'm happy to go back to a little bit the details of how we do that. Because it's a non-trivial step. So we obtain an overall motion, that is the overall diffusion coefficient of our particles, that is also given by an Einstein law, kT over an effective friction coefficient. And I give you the effective friction coefficient, which writes for just one leg in the following way. So it's a sum of 2 terms. The first term is related to the unbound states. It is the probability P naught that my particle is unbound over the friction coefficient gamma of the unbound particle. Then there's another term that is related to the bound states, where P1 is the probability for my leg to be bound, and the friction contribution to this bound state is some factor gamma plus a gamma prime. What is this gamma prime? Well, first of all, this means this friction contributing to the bound state is always greater than the base friction of my particle. And that makes sense because, my particle is now attached to the surface via the leg, and that makes it harder to move. And what goes in there? Well, all my parameters go in there. So in this little gamma prime, what is there? There's the hydrodynamic friction of the little spring that feeds into this bound state. And then there are recoil terms associated with the spring recoil force characterized by the spring constant K. And basically, you can think of it a little bit like Velcro. When you're bound with Velcro, it's very hard to move across the Velcro. So basically, my spring exerts a friction associated with being bound. And what I need to characterize these friction terms is a time scale. And so there are 2 time scales feeding in this effective friction when I am bound. One of them is the time that I am bound, which makes sense. And the other one is the time that it takes my spring to relax towards equilibrium. Okay, so we can check, that this formula works for, at least numerically, for just one leg. And then, of course, we want to extend that to multiple legs. So we add lots of legs. We can do the same description. We can use the same homogenization framework. And I think what is super nice is we obtain what I find an elegant mathematical formula, where the formula extends for the effective friction to a sum over all the possible states of my system, weighted by the probability to be in that state, the probability to be unbound, to have 1 bond, 2 bonds, etc., until n bonds, where n is the number of legs. And each time there's a specific friction coefficient contributing to that state. And we can show that this friction coefficient scales linearly with the number of legs that are bound in that state, which makes sense because, the more you have legs bound, the harder it is going to be to move. But a priori, it was not obvious, if the scaling was linear with the number of bonds or quadratic or whatever. Another reason why I like this formula is that you can here there's an analogy between this formula and resistors in parallel, for example. So here, what is subtle is that you're not adding the friction coefficients, you're adding the inverse of the friction coefficients together. And that's a quite subtle effect. But when you look at these gamma n's, they're actually adding the friction coefficients, gamma plus gamma prime. So there's this double tangle. Okay, so cool. I have this cool formula. What can I do with it? So we can write, for example, so we can gather up all these terms, and I rewrite here the effective diffusion coefficient that is predicted from our model. Now, can we calculate it? There are some parameters in there, like the friction coefficient gamma, the spring constant K, that are kind of well known, or at least that you can pretty well estimate from polymer physics. Okay, so that's so far so good. And then there are a few of them, like the number of legs that can potentially bind, like the strength of the bond itself, that are very, very hard to quantify. Sometimes you can estimate them and it's okay, but in our system of DNA coated colloids, where we could vary a lot the temperature, those two parameters would vary a lot. So we wanted to be very quantitative in terms of how we were understanding these things. And so we spent quite some time figuring them out. So without going into the details, I'd just like to say a few words on how we figured them out. So in that process, basically to figure them out, we had to understand how our particle was attached. So not how it was moving, but first how strongly it was bound to the surface. And what that meant was trying to figure out the free energy of interaction of my particle with the surface. And to do that, Fan Kui, who was a postdoc in Dave Pine's group, developed a total internal reflection microscope that basically was sending an evanescent wave inside our sample, from which we could detect where the particle was, and then reconstitute from that the distribution of positions of the particle, and then the free energy of interaction. So she could obtain, for example, free energy potential profiles with respect to height. And here I'm showing two curves, one with non-sticky DNA in gray, and one with sticky DNA in blue, that shows that there's a definite potential well close to the surface that corresponds to this sticky attraction. So of course, the setup is measuring the overall stickiness, but then doing an extra layer of modeling, we can understand how this overall stickiness is determined from microscopic parameters. So we did that. There's basically two kinds of forces that go into phi of h. There are volume forces, but mostly surface forces due to surface-surface interactions. And then it's just a zoo of interactions. We've got specific binding and unbinding, which is rather complex because there's entropic degrees of freedom in play. There's steric repulsion due to the fact that you're compressing polymer brushes, but then there's also van der Waals attraction forces because those surfaces are pretty close to each other. There's also electrostatics at play, and then depletion forces inside the sample. So it's a zoo. And we took some time to rationalize, which were the forces that were more important, what were their effects, and then how from these basic physical principles we could reconstitute the free energy of interaction. Okay, we did that, and basically we obtained pretty good agreements with experiments with just one parameter fit, and then we could change the temperature, change the substrate, the sample, and we obtained similarly a good agreement between experiments and theory. Okay, this very long detour to say that we have some understanding now of the microscopic attraction between the surfaces, which gives us, in this big formula, the number of legs and the strength of the interaction Q1 over Q off for each individual bond.
Sorry, I just had one one question. Maybe, I don't know if this is what you're going to talk about next, but in terms of the probability of being in each of these states, that's something that also, I guess, one would need to know to calculate this effective diffusivity.
Exactly. So the probability, for now, we took as an assumption that the probability to be in each of those states is determined by the ratio of rate of being bound and being unbound. So for example, if you have just a two-state model, it would be the ratio between Q on over Q on plus Q off. And therefore, if we can determine, we know Q on from sort of standard chemistry kinetics, but the ratio of Q on over Q off is much harder to measure because it depends on the temperature, the landscape of things. And so a priori, we can measure that ratio with, or we can infer that ratio from our microscopic model. That gives us a good agreement with the macroscopic measurement of free energy, which gives us also the probability for one individual state, for one bond to be there. And then we assume that if we have n bonds, the probability to be bound, those probabilities are independent for now.
Got it. And then I just had one other question too. Since the nature of these interactions is through, I guess, these single-strand DNA interactions,
Mm-hmm.
Can you connect the surface interactions in some way to just the kind of binding energy in a double-stranded DNA? Does that end up being kind of the dominant interaction energy?
Yes, so the dominant interaction energy is indeed, so in this Q on over Q off, right, a bit earlier, sorry, you're going to see my whole screen. I was very quick about that, but in Q on over Q off, you have, the strength of the bond. So if you were in a very simple system, it would only be the strength of the DNA going from single-stranded DNA to double-stranded DNA. But there's a little, that's the dominant term, but there's a little bit more that goes into play, and that's due to those entropic forces. It seems a little bit more, but in fact, the effect is really not small. So for example, the melting temperature of 3 DNA pairs in solutions would be around 90 degrees. But here the pairs we're looking at, they're more around 50 degrees. So those entropic forces, they make the bond weaker, and they make it weaker in nonlinear ways. So it's a very cool system for that reason. Okay. Thanks for the interruption. I was not necessarily going to answer that question. So we have some idea that we're pretty much putting in numbers inside these parameters that make sense. And so then we calculated our prediction for the effective diffusion coefficient in our system of DNA coated colloids and plotted it against the experimental data. And so the model is the black line and the experimental data are the points. And this is giving, again, the diffusion coefficient with respect to temperature relative to this melting temperature. So we obtained a pretty good agreement, and we were pretty satisfied with this. But what was more interesting to us is what the model was telling us about what was going on. And in designing the model, I said we had to figure out the energy of interaction with the surface, which means we had to figure out, basically on this picture, what was going on right at the binding point in terms of how many bonds are actually formed. So basically, what's the average probability of being bound, and so on and so forth. And from that, we were able to establish that the number of legs that can bind, and then eventually, of course, the numbers that are bound, really increases with temperature. As you lower the temperature, the particle gets closer and closer to the surface, which means more and more legs are available to bind. And because of this very big increase of the number of bound legs, which is proportional, sorry, number of available legs, which is proportional to, nearly proportional to the number of bound legs, you get, sorry, I'm going to jump, you get this very, very big decrease of the diffusion coefficient. So I'm sorry, my legend is a bit confusing, but actually in pink, this is the average number of bound legs, so not the available ones. The available ones increase even more. And you can see here the number of bound legs goes from about 1 to about 40. So it increases by nearly 2 orders of magnitude. And that's about the 2 order of magnitude decrease that we get in the diffusion coefficient, which comes from this linear dependence of the friction coefficient with the number of bound legs. So really, when you have more legs with the surface, bound to the surface, it's harder to move. A message that's quite simple.
And just in terms of the interpretation, this sort of pink graph that's showing the number of available legs basically going to 0 above this melting temperature, what's kind of the way to understand that? Because I guess I'm thinking about this hairy ball. It seems like there's even, when there's some higher temperature, there's still some legs that are kind of available to bind.
Yes. So of course it's an in average picture. And so in my model, I only showed this 1D view, but in fact, there's a little bit of 2D that you have to think about. So in average, at very low temperatures, the particle is really close, really tightly bound to the surface, really close to the surface. And at very high temperatures, it's free to fly anywhere. So in average, it's not near the surface. In average, its average position is in the bulk. So the average connections it can make is actually really 0. So there's this continuous transition between the two. Maybe that makes more sense. Okay. Okay, so we figured out, a little bit about how fast they move. And then we wanted to understand how they were moving. Am I doing good on time? I'm okay so far. Okay. There's when you read the literature, there's a variety of names and ways people have interpreted how such particles move. And of course, it's different from one particle to one particle. So there's a zoo of things they can do. They can hop, they can roll. For example, the white blood cells, some people say the viruses crawl or glide. Some say they slide, they hop, they jump. They can do lots of things. So we wanted to understand a bit more potentially what our particles were doing, and also quantify a little bit what that meant, what those words were meaning for us in our context. So looking a bit more at the experimental data, when we look closely at the trajectories of our particles, some trajectories look a little bit like this. So here you can definitely see there are like these patches and also these steps in between the patches. And in fact, we can go a little bit further and look at all the possible step sizes that the particle can do. So here I plot the probability to do a given step size with respect to the step size, and I plot it in semi-log scale. And for normal usual diffusion, you expect to see a parabola. And hopefully I don't need to convince you this is not a parabola. So there's definitely, short steps and then much longer steps. So that's just another way to quantify what we're seeing in the picture. So then I can at least do something to look a little bit more at what's going on. For example, I can color on this single particle trajectory the short and the long steps. This is what we've done here, for example, and we see something that is pretty hard to interpret, but that's, the short steps are confined and the long steps help you go from one region to the other. But overall, the long steps are everywhere on the trajectory, and the short steps are not essentially everywhere, but everywhere in the patch where you're observing. So what this means is, likely the particle is transitioning from these long steps to these short steps, but not necessarily due to the surface that it's exploring, rather because it's switching permanently between those two modes. Okay, so we can go a bit further than that, and to quantify a little bit what's going on, I'm going to use other words than short steps and long steps. I'm going to call these big jumps hops, and then I'll justify why we think they're hops, and then I'll call, the shorter steps. It's harder to put a name on the shorter steps, so they can be any form, a thing on the surface, either sliding, rolling, or crawling. And I will use for now crawling, but then I'll switch to something a bit more specific. Okay, we can go a bit further. One thing we can do is from this distribution of steps, we can model it by the sum of two Gaussians, which will tell us the probability to be in one mode or the probability to be in the other mode. That's the first step. So for example, the probability to crawl or to hop. And we can do that at all the temperatures that we're observing at our system at. And then here I will plot the probability to be in either mode. So in orange to crawl and in blue to hop, relative to the temperature, relative to the melting temperature. And we were super happy to see that there's a very big difference between the mode that is apparently most favored with respect to temperature. So crawling dominates at low temperatures and hopping dominates at high temperatures. So what's going on? So we can shed a little bit of light on this behavior by digging into the model that we built previously. And actually, we don't need to improve the model. In fact, the model can tell us how fast the particle hops and how fast the particle slides. And so it can only do these two things. It cannot do other forms of motion. The reason for that is that it's in 1D. And again, to be very specific on what I mean by hop and slide, I made little movies. So hopping is what happens when likely you detach all bonds from the surface, float in free space, and then reattach. And of course, this is diffusion, so it can go one way or the other. And then sliding is what happens when you keep at least one bond in contact with the surface and then move the other ones sequentially. Okay, so it's a form of walk on the surface. Okay, so what was in the model? Well, in this model, we had this big formula. And in fact, in this formula, there is the probability to hop and the diffusion coefficient while hopping in this unbound term. So one thing I can do is build the diffusion coefficient for hopping from this term that scales like some little number x that is smaller than 1 times the power n, where n is the number of legs. And then sliding corresponds to what happens when I have at least one bond in contact with the surface. So that corresponds to all these other terms. And that's n terms, but they focus on the term corresponding to the average number of bonds. And then I can quantify the probability to be sliding, but also the diffusion coefficient while sliding, which scales inversely with the number of legs n. As a result, I get two predictions, one for the diffusion coefficient for hopping, one for sliding, which have different scalings with the number of legs. One is exponentially decreasing with the number of legs, and one is only harmonically decreasing, which means that likely, if you have lots of legs, it's faster to slide than to hop. And the take-home message of the model is, well, hopping is what happens, is rather easy when you have just a few legs and specifically a few bonds with the surface. It's rather likely to unbind them all at the same time and then hop. But when you have lots and lots of legs, coordinating them to all detach at the same time is definitely a rare event. So you do pay a price by trying to slide, right? Because you're attached by all these tethers, but overall, it's likely a more efficient way of covering the surface. Okay, so you're really, walking at the nanoscale, like caterpillar, and you do that when you have lots of legs, like a nanoscale caterpillar. So for our systems, I showed this plot with the number of legs with the surface, and with temperature, the number of legs definitely changes a lot. So at high temperatures, you have just a few bonds, we're likely going to hop. And at low temperatures, you have more than 40 bonds, so you're more than likely to slide. So hence the difference between probabilities that we were seeing. And then we can go one step further and quantify how fast hopping or sliding is, and overlap it with our experimental data. So I overlap the diffusion coefficient for hopping in blue and the diffusion coefficient for sliding in orange, and we find pretty good agreement with the data. So we believe that at least there's part of this, at least that we have a partially good understanding of what's going on. Okay. I think, okay, I'm sorry, I will skip this. I hate people skipping, but I will, okay. I'm soon going to conclude, but I wanted to finish starting again where I started, which was, can we understand, what's going on in terms of nano-caterpillar motion? And then the next step is, can we control a little bit what's going on? So understanding, we've seen how fast that it slows down significantly with lots of legs, and then that's due likely to different modes of motion, either hopping or sliding or some form of cohesive motion on the surface. But can we go one step further? And when you think a bit about it a bit more, when I showed, for example, these equations, there are all these parameters that come in. And those parameters, they depend on, how many sticky ends I've put on my surface, so how many legs I have, of course, but they also depend on the polymer properties, so the length of the legs, but also, the strength of the molecular bond, so I can choose different bond strengths. And overall, this means that I have very different ways to change whether or not I'm hopping or sliding, or what I'm doing and how fast the particle is moving. And with our model, we established in this context a sort of phase diagram for caterpillars, which has two axes. On the lower axis, you have the strength of the bond. So a very strong bond is towards the left, and a very weak bond is towards the right. So they're very sticky towards the left. And then on the vertical axis, you have some form of relaxation rate that tells you basically how wobbly your legs are. So you can have very stiff, very short legs at the top, or very wobbly, very wiggly legs at the bottom. So very long polymers. And then this chart divides the space in two, in hopping and sliding. And so what's going on? So for example, to understand why you're more doing one than the other, well, for example, at the bottom right, you're definitely sliding because your bonds are pretty weak and your legs are wobbly. So moving is pretty likely, but then, detaching and reattaching is pretty likely because your bond your bond strength is pretty low, but also you have very wobbly legs. So the friction coefficient you pay trying to move is much lower. So what can we put on this chart? We can put DNA-coated colloids that I've talked a lot about today, and around their melting temperature slightly lower, most of them are sliding. The smallest ones are a little bit in between. But I've talked about other particles, and one of the particles I've talked about are viruses. And what's cool is, especially with I shouldn't say that, but especially with the coronavirus crisis, we have lots of data on coronavirus, for example. So it's quite easy to put coronavirus on this chart, and also other viruses. And viruses end up all the way up there. What happens is they have very, very strong bonds that are long-lived. It's about 1 second, the rate of attachment and detachment of the spike protein. And they have very stiff legs. The spike proteins are very stiff. So what this tells us is, likely those particles, they won't slide, they won't do a form of coordinated walk on the surface, they can only hop. And in fact, within our framework, and in fact, for example, the experiments that were done in influenza A, those jumps that were called gliding were definitely observed. Now, viruses don't just hop. And indeed, they found other strategies to move. For example, they can roll, but they can roll by active cutting of the bonds that have been formed. So which is an out of equilibrium form of motion that is, for example, a perspective of these work. I am done for now. I find nanocaterpillars fascinating things, fascinating objects, and that strike a lot of fundamental questions on how they move in a nice context. So, for example, here I show just this picture of the work by Nancy Ford, where she's designing artificial motors or artificial colloidal motors that move by cleaving their bonds and rolling on surfaces by sequential binding and unbinding. And there's lots to understand on how they move. And then I mentioned some of these, but there's lots of technical issues in trying to understand this motion. We've had, we built several experiments that were from large scale to much smaller scales. There's questions of how you design simulations that do these kinds of things, and so on and so forth. And then what I like is there's lots of applications in this context, some that I talked about and more in various contexts. This crowd is not very big, but I don't know who's out there. So I'm advertising that I have a fully funded PhD position in my group, and we're going to try to observe soil dynamics in soils. There's lots to unravel, and this is going to be a really cool PhD project. So please reach out if you want more info. And finally, stop here. Thank you so much.
Thank you, Sophie. Do you have any questions?
One thing I wanted to ask, so I guess here we're talking about this, both modes, this hopping and sliding, they're sort of diffusive modes and there's no directionality. So yes. Have you thought about sort of the maybe minimal versions of this or the changes that would be required if you wanted to model something directional? I guess in my head, I'm thinking of like a tardigrade or something that's like crawling around in a kind of directed way.
It's so there are several ways that it can do some form of be directional. Targeted is another word, but directional comes up if you have you need a breaking of symmetry, so you need a little force likely. So for example, white blood cells, they're definitely moving in one specific direction. It's the direction of blood flow because they're pushed by blood flow, basically. And already there, it's not clear how this all works out because you get forces on your springs. You might get lots of nonlinearities. I don't know what's going on. But then just another way you could think about this is, for example, patterning your surfaces with more or less sticky ends, and then hoping that your particle would, preferentially follow that track that maybe has more sticky ends. That's not obvious, and it's not obvious that it works because you always have this trade-off of when there's less sticky ends, you explore faster. When there's more, you stick there, but you explore slower. So it's not clear how they deal with heterogeneity or different patterns in space. And in fact, this may be leading to kind of odd behaviors at low temperatures that I have not mentioned too much here. And mathematically, it's a very hard problem too. That's what I can say. One of the hard issues is that now you have to add binding rates that really depend on space, and that's not something that's easy to model and that you can do like that. It's lots of assumptions, and then it makes the math behind much harder.
I see. Thanks.
Is there any other biological examples of directional movement other than red blood cells?
So white blood cells, so molecular motors are fundamentally out of equilibrium, but on long time scales, molecular motors diffuse, but on short time scales, they only walk along one line. Viruses, especially influenza A that has bond cleaving, it has, a persistence direction in one direction when it cleaves behind, because it cannot go back where the bonds have already been cleaved. Who else do we have? Bacteria some bacteria also have these kind of adhesion forces mediated by ligands. They can be also pretty directional, but I don't know if that is due to their multivalent adhesion or something else. Yeah, those are the examples that come up in biology. And then there's one big example of directionality, and which I haven't talked too much about, slightly smaller scales, which is the nuclear pore complex, which is a channel with lots of polymer brushes, and particles going through can bind to those brushes and then translocate. And this pore has specific directionality. Some things go through this way, some things go through that way, and nobody knows why.
Oh, is that right?
Well, there we have clues, but it's a big challenge.
Wow, it's so cool. It's such a fundamental biology, but I guess we need people like you. Okay, do you have other questions? Right, if not, let's thank Sophie again.
Thank you for your attention. Have a nice end of semester, since I was the last one.
Thank you, Sophie.
Thank you so much. Okay, bye.
Bye.
See you, everyone.
Let me know, Thomas.
Oh, yeah, I will. You'll be hearing from me.
Perfect. Bye.
Bye-bye.
Have a good day.